Hello everyone, hope everyone is doing great. This is Dr. Tehrani. Gonna do an exam review today for our upcoming exam on Tuesday, which I will post. It's gonna be 40 multiple choice questions that I'm gonna review in a few seconds. Gonna be 10 questions from chemistry, 10 questions from cells, 10 questions from tissues, and lastly 10 questions from skin. Okay, let's before I jump into the exam review, uh, I'm gonna give you references as I go through the questions. I will give you references on what pages to study. It's gonna be three documents that I'm mainly gonna refer you to. First document is gonna be the lecture notes. Second document is gonna be the supplemental notes. And on a very few questions, I'm going to refer you to a specific pages on the text reading. So all those are posted, uh, and you can have them open and as I'm going through these questions, make sure to take notes, and then you can go back and listen to it again as well, and then go back to the pages that I'm gonna reference you to and study those paragraphs that I'm gonna tell you. Okay, let's go ahead and jump in and get us started. This is the first time that I'm doing this. I'm doing an exam review. I usually don't do an exam review, and what I would usually do is give you a lot of time on the exam. Usually for 40 questions, I would give you about 400 minutes to complete it, and then I would curve the grades. However, with this now, with me doing an exam review, I might cut back on the exam time. I'm still debating on that, whether I'm going to cut it down to maybe 100 minutes for 40 questions or about 80 to 100 minutes for 40 questions, because I believe if you listen to this exam review that I'm going to do right now and take proper notes and follow everything that I say, you're going to do great on the exam, because there is not going to be a question outside of these questions that I'm going to review for you today. Okay, all the questions are multiple choice. Exam is going to be posted on Tuesday morning. I'll post it probably Monday night. I know this is a last minute video. I'm really sorry about that. And multiple choice questions, 40 questions, 10 questions from each section. And they're pretty straightforward. Only one attempt. There is no lockdown browser, because all my questions, no matter how hard you try to Google them or put them in the Google search bar, copy and paste it, you're not going to be able to find the questions. I've made sure of that. I've specifically picked questions and went after questions and put together questions that you won't be able to find on Google platform, because a lot of these days, when you're attempting an exam, you can easily just copy the question, paste it in the Google search bar, put two quotation mark, and then, believe it or not, the question will pop up and you will see the correct answer. But none of these 40 questions are going to be found on Google, so they are very exclusive. All right, let's start. I'm going to be jumping around between chemistry, tissue, cell, and skin, but I will tell you. So the first question that I have asked, the first question, it's going to be a chemistry question, and it's about nucleus of the atom. So nucleus of the atom, basically you need to know what an atomic number is, because the question is about nucleus of an atom and atomic number. So to study for this question, go to page 2 of your chemistry lecture notes and study objective B. Again, page 2 of chemistry lecture notes, study objective B, look for atomic number, and definitely read the paragraph that talks about nucleus of the atom. Second question is again chemistry question. It pertains to the energy level of the shell, the atomic representation of the energy level of the shell. Basically, it's asking about how can we have a stable atom. And if you have read through, this is going to be on page 2 of chemistry lecture notes, section 2.2. So read through section 2.2 of page 2 of chemistry lecture notes. It's going to talk about how you can achieve of having a stable atom. Basically, it's going to go over the shells. Remember, to have a stable atom, it usually the first shell can only occupy 2 electrons. Okay? So if you have one shell only, and the first shell only, and the first shell, for it to be stable, it needs to have 2 electrons on it. Can go to a second shell based on number of electrons. In order for it to be stable, it would need to have 2 electrons on the first shell. The first shell can only accept 2 electrons. The second shell can accept up to 8 electrons. So if the atom that you're looking at has the first shell with 2 electrons and a second shell that has 8 electrons in it, that's a stable atom. If you have an atom that only has one shell with 1 electron, it's not a stable atom. If you have an atom that has 2 shells, the first shell is filled with 2 electrons. However, the second shell, let's say, has 6 electrons. That's not an stable atom. It can go up to another shell as well. It's called the third shell. That can also have up to 8 electrons. So for an atom to be stable, the first shell has to hold 2 electrons, the second shell has to hold 8 electrons, and the third shell has to hold 8 electrons. Again, this depends on the number of electrons. If you only have an atom that has 2 electrons, well, the first shell is going to be filled and it would be a stable atom. If you have an atom that has 18 electrons, you would put 2 electrons on the first shell. Second shell, you would have 8 electrons. And then the third shell, you would have another 8 electrons. That's a stable atom because you have filled all the shells. So again, read page 2 of chemistry lecture notes, section 2.2. It's going to go over what I just went over. Also, you can read page 7 of chemistry supplemental notes. It also goes over the same concept. Next question is about you need to know the composition of the structure and the composition of carbohydrates, proteins, lipids, and nucleotides. And you need to know the ratios of their composition, like how many for a carbohydrate, how many carbon to a hydrogen to an oxygen. For example, let me give you an example on this. Sugar molecules, they have, let me turn down the TV a bit. Sugar molecules usually contain, what are the 3 that they contain? Carbon, oxygen, and hydrogen, right? Carbon, oxygen, and hydrogen. And my question is going to be, what is the ratio of these 3 going to be? So carbon, oxygen, hydrogen, it's going to be in a ratio of 1, 1, 2. As to 1 carbon, 1 oxygen, and 2 hydrogen. For example, the formula for fructose, which is a sugar, and sugar falls under what category, guys? carbohydrates, proteins, lipids, or nucleotides. Sugars, they fall under carbohydrates. So, carbohydrates, carbon, hydrogen, and oxygen. What is the ratio? 1, 1, 2. 1 carbon, 1 oxygen, 2 hydrogen ratio. Example, a structure or formula of fructose. C6H12O6. If you simplify that, that's going to be basically C1H2O1. So definitely know the ratio. There is going to be a question on that. Know the composition, mainly specifically for carbohydrates, but definitely know the composition of proteins, the structure of lipids, and the structure of nucleotides. This is in the chemistry section. Next question that I have picked is about a structure of ATP. A structure of ATP, this is very simple. I'm going to refer you to the section on page 13 of Cell Supplemental Notes. Page 13 of Cell Supplemental Notes. So the structure of ATP, there is going to be a question asking you what is the structure of ATP. So if you go to this page, you're going to see the structure of ATP. There is a ribose sugar. There is a base. The base is adenosine. And then on the other side, there are 3 phosphate groups. So remember these 3. If I ask you about the structure of ATP, what does it contain? What's the structure of ATP? There is a ribose sugar, there is adenosine as the base, and there are 3 phosphate groups. Next, it's about atomic bonds. So definitely be familiar with hydrogen bonds, covalent bonds, and ionic bonds. Don't go into too much details on them. All I'm going to ask about this is going to be basically definitely know which one is the weakest of them. Which one is the weakest of them? Hydrogen bond or covalent bond or ionic bond? I'm going to refer you to page 3 and 4 of chemistry lecture notes for this. And also page 3 and 4 of chemistry lecture notes is going to be actually section 2.5. And also page 4 and 5 of chemistry supplemental notes. So I refer you to 2 sections for this question. First section is page 3 and 4 of chemistry lecture notes, section 2.5. Next one is page 4 and 5 of chemistry supplemental notes. Next, again, it's about ATP also. What's the role of ATP in cellular energy metabolism? I'm just going to refer you to this page. It's page 13 of Cell Supplemental Notes. And also, I posted a PowerPoint in this section. It's under week 1 PowerPoint. Week 1 we covered the cell. Covered the chemistry, sorry. So, a slide 65 of chemistry PowerPoint. And that's posted under week 1 materials. I know I haven't posted a lot of the PowerPoints. What I'm going to do is starting this week, week 5, I'm going to start posting my PowerPoint slides. Everyone says, my previous students have said it's very helpful, and I think it's going to be helpful to you guys, especially some of you have problems reading through paragraphs, and I do understand my lecture notes are a bit quite few pages. Same thing with the supplemental notes are quite a few pages, and then I post the text reading as well. I do not want to overwhelm you. That's why I have these exam reviews. And always, if you have any questions, if you know how to start, where to start, which document to start with, email me and I'll send out weekly emails. For example, this week we are covering week 5. We are covering the muscular system. I highly encourage you to start with the muscular anatomy PowerPoints that I have posted, the slides, and then follow that with the muscular physiology slides. So I have 2 different slides, 2 different sets of slides. One slides are the muscular anatomy, which covers the anatomy of the muscular system, and the other set of slides are the muscular physiology, which covers the physiology section of the muscular system. Now, a lot of my questions for lecture are going to be physiology questions. And a lot of the questions, over 99% of the questions on lab quizzes are anatomy questions. So the anatomy part basically focus that for the anatomy quizzes, the lab quizzes that we have. I'm not going to ask any physiology questions on lab quizzes. All lab quizzes are identification. All of them are you have to identify the structure and definition. I don't ask any processing questions or physiology questions where you have to think about the sodium potassium pump and etc. When we're gonna go into the nervous system, there's a lot of physiology in the nervous system, a lot of physiology in the muscular system, and a lot of physiology in the respiratory system. I don't think we covered respiratory system. We covered that the second. We covered it in another course. Okay, so sorry, I went sidetracked. Back to this. Role of ATP in cellular energy metabolism. And I'm going to refer you to page 13 of Cell Supplemental Notes and also slide 65 on Chemistry PowerPoint, which is posted on their week 1 material. The Cell Supplemental Notes is on their week 2. However, the Chemistry PowerPoint is on their week 1. So definitely for this question, if you study slide 65, you'll be able to get it right. Okay? Next question. I'm gonna be asking about we are still in chemistry, by the way. Difference between atomic number, mass number, and what isotope is. So, definition. If you understand the definition of atomic number, definition of mass number, and what an isotope is, you'll be able to answer this question. I'm going to refer you to page 2 of Chemistry Lecture Notes, Objective B, Section 2.2 and 2.3. I'm going to say that again. For this question, where I'm going to ask you to identify the difference between atomic number, mass number, and what an isotope is, I'm going to refer you to page 2 of Chemistry Lecture Notes, Objective B, Section 2.2 and 2.3. Next question, which is one of favorites of mine, and a lot of the students get this question wrong. And it's a very simple question. I think it's a very simple question. It's just a matter of understanding the definition of osmosis. The question is going to simply ask you, what's the definition of osmosis? Remember, write this down. Definition of osmosis it should always include water. Basically what it is. So don't get thrown off with other answers that you're going to see A, B, C, D. Look for an answer that has water in it. Because what it is movement of water through a semi-permeable membrane from a more dilute solution. What is a more dilute solution? A more dilute solution is a solution that has less concentration. Okay, sorry about that. Oh no, my computer. Because I didn't move the mouse, I think it went to sleep or something. Okay, going back to osmosis. Definition should include water. I'm going to go over the definition. It's basically movement of water. It's not movement of anything else. It's movement of water from through a semi-permeable membrane from a more dilute solution. A more dilute solution would be a solution that has a less concentration to a solution of a higher concentration. To a solution of a higher concentration. That would be a solution that's less dilute. So remember that. Again, I'm going to refer you to a few pages to fully understand this. It's going to be pages 18 and 19 of Cell Supplemental Notes. Page 18 and 19 of Cell Supplemental Notes. Okay, next. Next is going to be diffusion. Know the process of diffusion. I'm going to refer you to a few pages for this question. Page 2 of Cell Lecture Notes, section 3.4, and page 16 and 17 of Cell Supplemental Notes. Go to those pages and look for the definition of diffusion. Okay? Know the process of diffusion. Again, page 2 of Cell Lecture Notes, section 3.4, and page 16 through 17 of Cell Supplemental Notes. Next, osmosis, diffusion. Okay. Features of filtration and process of filtration. And mainly how it differs from diffusion. So, we talked about osmosis. One question on osmosis. Next question is on diffusion. Next question is on filtration, or the difference between filtration and diffusion. I would refer you to pages 21 of Cell Supplemental Notes. Page 21 of Cell Supplemental Notes. Next, we're going to talk about plasma membrane. We are in the cell now. Again, some of the question is not like the first 10 are chemistry, the second 10 are cell, and the next 10 are tissue, and the last 10 are skin. No, they are all mixed together. So that's why I'm jumping around. Next one is going to be, as I said, after filtration. Next question is going to ask you about the composition of plasma membrane. Composition of plasma membrane. Very important question. This is very important for you to know. That's why I'm going to refer you to quite a few pages on this and sections. First, I want you to go to page 2 of Cell Lecture Notes, section 3.2, and read through that paragraph that talks about the composition of plasma membrane. Basically going to be about the phospholipids and hydrophilic tail, hydrophobic tail, how they are structured, and which end is where. Also, For the same question, I'm also going to refer you to page 4 and 5 of cell supplemental notes. So, page 2 of cell lecture notes, section 3.2, page 4 and 5 of cell supplemental notes. Should give you a beautiful, clear understanding of composition of plasma membrane. I promise you, if you read those two sections, they are not long. I'm giving you sections. I'm not giving you pages to read. These are like small paragraphs that I'm giving you to read. You're gonna do wonderful. You're gonna get this question right. Okay, next question. You need to know the different types of transports. Different types of transports. What do I mean by that? Different types of transport. What are the different types of transport? Osmosis, diffusion, passive, which is also facilitated transport, and active transport. There is a very simple question on that, and I'm going to refer you to page 14 through 16 of cell supplemental notes. Also, page 2 of cell lecture notes, section 3.4. Again, this question is asking you about the different types of transport. For example, it might be, what do I mean by that? It's a very simple question. Which one requires energy? Which one doesn't require energy? And I'll give you 4 choices: osmosis, diffusion, passive, active. So to better understand this question and to be prepared for it, I'm gonna say it again. Page 14 through page 16 of cell supplemental notes. Also page 2 of cell lecture notes, section 3.4. Okay, next question. Very important question. I always include this question on all my exams, and a lot of people do not get it right. It's about sodium potassium pump. It's about sodium potassium pump. It's not difficult. It's very straightforward. It's very general question about sodium potassium pump. We're going to talk more about it later on down the course, but for now, when I say sodium potassium pump, it can also be called N, which is for the sodium, and then dash K, which is for the potassium, NK pump, or sodium dash potassium pump. What I need you to know is function of sodium potassium pump. So I'm going to refer you to some paragraphs and some sections. First section I'm going to refer you to is page 28 and 29 of cell supplemental notes. Oh, now we're getting to the text reading. Also, page 17 of cell text reading. So when you open that PDF file on the cell text reading, the PDF, I mean, if you're opening it with the Adobe and anything else, it has like 1 out of 25 pages, for example. However, when you scroll down, you see there are also page numbers on each page. Like page 1 corresponds to page 75 that's typed on that page. So I'm going to give you both page numbers. So when you open up the cell text reading to a study about the sodium potassium pump, I want you to go to page 17, which also corresponds to page 93 of the document. Page 17 is the page that's shown on top of the preview. I use Mac, so mine is a preview, but if you use PDF opener, it will have like pages 1 through 25. However, on each page of the document, because that's extracted directly from the text, it has the textbook page also. So these, when you open that cell text reading, go scroll down to page 17, which corresponds to page 93 of the text reading. And when you go to the text reading, page 17 corresponding to page 93, I want you to look on the lower right hand side. Lower right hand side where it says the sodium potassium pump helps maintain cell volume. It's a paragraph, read it, understanding, and you get the question right. And then continue on to the next page of the text, which is page 18, corresponding to page 94. Okay. We are only on question 14, guys. Question 14 which is about tissue. Definitely know the components of connective tissue. Components of connective tissue. This question has been taken directly from page 17 of Tissue Supplemental Notes. Directly from that question. Directly from page 17 of Tissue Supplemental Notes. It says components of connective tissue. Learn them, memorize them. Next. Okay, next is going to cover about 4 or 5 questions about tissues as well. I need you to know where you can find the location, of different types of connective tissue. The main locations, okay? The common locations. Areolar connective tissue, adipose connective tissue, reticular connective tissue, dense, fibrous, regular connective tissue, dense, irregular, hyaline cartilage, elastic cartilage, and fibrocartilage. So the question is going to be, what location can you find? Which of the following location would you find fibrocartilage? Which of the following would have reticular connective tissue? And these are, you can find the answers. These are in a table, actually, very easy, very straightforward. Pages 16 through 35 of Tissue Supplemental Notes. Pages 16 through 35 of Tissue Supplemental Notes. If you don't want to go through all those pages, here's another table that I'm going to refer you to. Table 4.5 on page 7 of Tissue Lecture Notes. Very helpful. Very, very helpful. What I'll be mainly asking is location of types of tissue. And a lot of the locations of those types of tissue you can find in table 4.5 on page 7 of Tissue Lecture Notes, guys. Okay, I'm going to go to the next question. Know the types of simple epithelial tissue and locations of each. Very similar to the previous question. Previous question, I was asking, know where you can find the location of different types of connective tissues. Now, I'm asking you to know the types of simple epithelial tissues and location of each. Simple squamous, simple cuboidal, simple columnar, simple ciliated columnar, pseudostratified columnar. Now, I will not ask you to identify any of them. That's for lab. When I give you a structure and ask you which one is which, when you're identifying the type of the tissue, that's for lab, that's not lecture. I'm not going to ask you that question on lecture exams. Again, I'll mainly be asking where they are commonly found. Okay? In the lymph, in the kidney, in the bladder, you know? Also, you need to know the types of stratified epithelial tissue and location of each. The previous one was types of simple epithelial tissues and locations of each. Now the next one is types of stratified epithelial tissues and location of each. We have keratinized stratified squamous, non-keratinized stratified squamous, stratified cuboidal, and transitional epithelium. Transitional epithelium. Well, that's a question. Now, let me refer you to some pages so you can easily find this. Pages 2 and 3 of tissue lecture notes. Pages 2 and 3 of tissue lecture notes, tables 4.1 and 4.2. All the answers are there. So study those tables. The table on the left side of the table, you're going to see the type of the tissue, and all the way on the right hand side, you're going to see the location of that tissue. Okay, jumping back, going to talk about chemistry again. A structure of proteins. Be familiar with the structure of proteins. I highly recommend that you study pages 20 through 21 of chemistry text. Again, this is the text now. Pages 20 to 21 of the chemistry text reading. Page 20 refers to also page 70. As I explained again, when you open up the text reading, on the top is going to be the pages from how many pages I have selected, and when you look at the document itself, on every page it has a different page number. I don't want you to get confused by that, so that's why I'm giving you both page numbers. So when you open up the chemistry text reading, I want you to go to page 20, which corresponds to page 70 on the document, and also go to another page down, page 21, which corresponds to page 71 on the document. Read those two pages. It's like two paragraphs that talks about the structure of the protein. Very important. If you read that, if you understand that, you're going to get the question right. Next question. Okay, next questions are pretty easy. Again, we're talking about the cell, the functions of organelles. You learned this in high school. Repetition. And be familiar with the functions of ribosome, rough ER, smooth ER, Golgi, mitochondria, and lysosome. I'm not going to ask every single one of them. I'm only going to pick two. So that's that. Next one is going to be function of phospholipids in plasma membrane. Remember, there was a question about plasma membrane. Now this question is about the phospholipids in plasma membrane. So for this, I'm going to refer you to the question is going to ask about the function. What is the function of phospholipid in plasma membrane? Okay, and you can find the answer. You go to page 2 of Cell lecture notes. Page 2 of Cell lecture notes. Also, page 14 of Cell supplemental notes. On page 2 of Cell lecture notes, go to section 3.2. Sorry, I didn't say that. So, again, functions of phospholipids in plasma membrane. I'm going to give you 3 segments to read. The first, page 2 of Cell lecture notes, section 3.2. Next, page 14 of Cell supplemental notes. And lastly, page 12 or corresponding to page 88 of Cell text reading. It's going to be when you open the Cell text reading and scroll down to page 12 or 88, which is on top of the document. On the lower right side, there is a segment that talks about where it says plasma membrane is lipid bilayer. Read that paragraph. Okay. Next is gonna be different types. Definitely, here's another question that's about tissues. Different types of specialized cell you can find within the epidermis. What kind of Okay, the first question. This question is going to be confusing a little bit because I'm asking you different types of what type of a specialized cell do you find within epidermis? Next thing you need to do is to ask yourself what kind of tissue is epidermis. So identify what kind of tissue epidermis is and then ask yourself what type of a specialized cell do I find in that specific tissue? Now, to help you with this question, I'm going to refer you to page 4 of the Skin Lecture Notes. Page 4 of a Skin Lecture Notes, section 5.11. Also, I'm going to refer you to page 3 or corresponding page 155 of a Skin Text Reading. So when you open text reading, a Skin Text Reading, scroll down 3 pages, and on the left side you're going to see it's also page 3 is also page 155 of the Skin Text Reading. And then I want you to read the epidermis section. It's about a paragraph. Make sure you read it. Read the epidermis section. Also, I want you to read page 6 of a Skin Supplemental Notes. So for this question that I'm asking about what types of a specialized cell you can find within the epidermis and what kind of tissue is epidermis, I have given you 3 references to go back to and study. Again, these references are very short. Don't think they're like 2 pages or 4 pages. They're paragraphs. First one, page 4 of a Skin Lecture Notes, section 511. Second one, page 3 corresponding to page 155 of a Skin Text Reading. Read the epidermis section. And lastly, page 6 of a Skin Supplemental Notes. Okay, next question is going to ask about a structure of plasma membrane and what it contains. You see, I've asked a lot of questions about plasma membrane. It's a very important concept. You need to know it well because it's going to carry on, and I want you to have a good understanding of it. That's why there are a lot of questions. All the questions are carefully picked and chosen because these are, I don't want to ask you questions, I don't want to trick you guys, I don't want to ask you non-irrelevant questions, difficult questions. Every question that I pick it carefully. I put a lot of thoughts into it. How is it going to be useful to you? And these are good questions. Okay. So this question is about the structure of plasma membrane and what it contains. I want you to refer to page 12, corresponding to page 88 of cell text reading. When you go to that page, when you go to page 12, open up the cell text reading, scroll down to page 12, corresponds to page 88 on top, and look at figure 3.13 and read the figure description. The question comes from the figure description. Figure 3.13 on page 12 of cell text reading, corresponding to page 88. Okay? If you read that underneath the figure, it's about a small paragraph description. You'll get the question right. Halfway through, guys. Halfway through. Okay, next question is about characteristics of epithelial and connective tissues. Characteristics of epithelial and connective tissues. Basically, I want you to understand, I want you to differentiate between the two. I want you to differentiate which one is closely packed and has little intercellular space. Is it epithelial or connective tissue? Which one has scattered cells with large amounts of extracellular fluid? Connective or epithelial? If you know the answer, you don't need to go to the page that I'm going to refer you to. Good job. That should be enough. However, if you're not sure about the answer, if you do not know which one is closely packed and has little intercellular space and which one has scattered cells with large amount of extracellular fluid, I want you to go to page 5, correspondent page 117 of tissue text reading. Page 5 of tissue text reading. Page 5 of tissue text reading corresponds to page 117 as well. Next question. Very easy. Basic functions of the skin. Basic functions of the skin. This is a giveaway. Page 1 of a skin lecture notes, objective B. Okay? Next. I want you to be able to list layers of a skin, especially layers of epidermis. I want you to be able to list layers of a skin, especially layers of epidermis. For this, I would refer you, it's very easy, layers of epidermis, know which one's the top, which one's the bottom. Page 2 of a skin lecture notes, objective C. Page 2 of skin lecture notes, objective C. Okay, next, we're still in tissues. Different types of muscle tissues. We have a skeletal, cardiac, and a smooth. I want you to know which ones are under conscious and unconscious control. Okay? And I also want you to definitely know the description of cardiac muscle. Okay? What makes it unique? What is so special about cardiac muscle? Intercalated discs. Okay? And I want you to remember, cardiac and smooth muscles are under unconscious control. They are automatic. Okay? You cannot control it like a skeletal. A skeletal, you can control it. The smooth muscles, smooth muscles, or under unconscious control, or they're automatic. For this, I would refer you to page 8 of Tissue Lecture Notes, Objective E, and also page 38 to 41 of Tissue Supplemental Notes. Gives you a better understanding of different types of these muscle tissues and explains what I just explained. So, page 8 of Tissue Lecture Notes under Objective E, also pages 38 through 41 of Tissue Supplemental Notes. Guys. Okay, next. Gonna ask a question about this. Can't leave this out. Know the description of nervous tissue and what are its distinguishing features. Distinguishing features of nervous tissues. Very easy, very simple, very simple. Let me refer you to page 42 through 43 of Tissue Supplemental Notes, as well as page 9 of Tissue Lecture Notes, section F. So read those two small sections and you should be golden about the question that I have asked about nervous tissue. Okay, next, I want you to be able to differentiate between dermis and epidermis. Differentiate between dermis and epidermis. Best way, I would recommend initially open up the skin text reading, go to page 3, corresponding page 155 of a skin text reading. Top left hand side, there is a paragraph, read that paragraph. Also, page 7 of a skin lecture notes, Objective F. Either one. If you read the text reading that I just referred you to, open up the skin text reading, scroll down to page 3, which corresponds to page 155 of the text, top left hand side, read that paragraph. Has the answer in there, has the answer and the question in there. You should be good to go. However, I'm giving you also another reference, which is page 7 of a skin lecture notes, Objective F, to help you with this question. It's a pretty easy question. And next two questions, I'm going to combine them together, are about, one of them is about sweat glands. So for sweat glands, read page 9 of skin lecture notes, section 5.27. Also read pages 8 and 9 of a skin supplemental notes. Know the structure and function. Know the structure and function. And the next one is sebaceous gland. The structure and function as well. Go to, for sebaceous gland, page 8 of a skin lecture notes, section 5.26. And page 9 of a skin supplemental notes. Almost done. 3 more questions. Okay. Homeostasis, very important. Favorite topic of mine. I believe it's one of the most essential concepts that you have to learn. And you will see these questions, especially the homeostasis questions, on final exam as well. That's how important they are. They are vital part of understanding anatomy and physiology. So, question that I have asked about Homeostasis is a definition question. So I want you to refer to pages 2 and 3 of chemistry supplemental notes and read the definition of homeostasis. I'm going to give you a great definition of homeostasis, which is even better than the one that you're going to read from page 2 and 3 of chemistry supplemental notes. It's basically, what is homeostasis? Your body's autonomic tendency, the body's autonomic tendency to maintain a constant internal environment, like temperature, cardiac output, blood glucose, blood pH. Wants it to be all the same across the range. Okay? As soon as something fluctuates, there goes in negative feedback and positive feedback. Positive feedback is not very common, but we have negative feedback that's very common. So, next question that I have asked after definition of homeostasis is, what is negative feedback then? I refer you to a page 3 of chemistry supplemental notes. However, I'm going to give you another definition myself. Negative feedback basically means that the body's response opposes the stress. What is the stress? So let's say you're out running and your body temperature, guess what? What happens to your body temperature when you're out there running? Body temperature increases, right? We call it the hyperthermia. It increases above 38 degrees Celsius. So negative feedback would kick in, and the response of negative feedback basically would mean that it's going to be the opposite. It's going to oppose the stress. So if your body temperature is going up, okay, and your hyperthermia, what's going to be the opposite or the negative feedback? Wants to bring it down, right? How would it bring it down? You're going to start sweating profusely. You're going to start sweating profusely. What happens when you sweat profusely? You cool down the skin. Same thing with blood sugar. What happens when your blood sugar goes up? Body releases insulin. Blood sugar goes down. Right? Same thing with a lot of different things as well. Blood pH. We're going to talk about this. This is more complicated. We're going to talk about it later on during the course about blood pH. But it's going to be hyperventilation, hypoventilation to get to the right blood pH. Now, you might ask, how about positive feedback? Positive feedback doesn't happen very often. What positive feedback is, it basically reinforces the stimulus, making it greater. Okay? Reinforces the stimulus and makes it greater. The best example of positive feedback is when a woman goes into labor. Hyperventilation, so the baby can come out. So it's not the opposing effect on the body. Okay? So definitely no. Question that I'm going to ask, definition of homeostasis, which I went through, body's automatic tendency to maintain a constant internal environment, like the temperature. And then the second question I have asked is about negative feedback. It's very common, more common than positive feedback. You don't see positive feedback very often. It means that the body response opposes the stimulus. So it goes the opposite direction of the stimulus. What is the stimulus? Can be the cardiac output, can be the body temperature, can be the blood glucose. So it goes the opposite. Okay? Last question, guys. Last question. Characteristics of layers of epidermis. Characteristics of layers of epidermis. Very simple question. I'm going to refer you to page 4 of the skin lecture notes, table 5.1. I'm going to ask 2 questions on this, and if you read table 5.1 on page 4 of skin lecture notes, you're going to do great. You're going to get the questions right. Hey, if you have any questions, concerns, comments, please email me, shoot me an email. I'm very responsive to emails. I want all of you to succeed in this course. I know with COVID, we have to make things work out, and it's not same as being in the classroom or me doing a lecture. I wish I could do that. I feel like the quality of the education is not the same, but I'm doing my best, and I wanna give you all the best as well. So if you need more information, uh, more lecture notes, or more documentations, any questions, reach out to me. We do have a tutoring session. I highly do not think any one of you would need a tutoring session because you're all doing so well, and I'm sure you're going to do great on this exam. Rewatch this exam review session that I just did. Write down the pages that I told you, that I referred you to, on the text reading, on the lecture notes, on the supplemental notes. And for me, just as I was preparing, it came out to be literally for 40 questions. It's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 15, 16, 17 pages. And I know this recording is almost an hour. It's over an hour now. And right now it's 10:42 p.m. on Monday. And I have promised to make the exam available tomorrow, but that doesn't mean that you have to attempt it tomorrow. I'm going to leave the exam open until next. I'm thinking, because I'm posting this video so late, I'm going to leave it open until next Tuesday, guys. So the deadline for the exam, which I'm going to post tomorrow, is going to be next Tuesday, which is going to be September 29th at 11:59 p.m. Okay? So no rush. Don't think because I have posted the exam tomorrow that you have to take it tomorrow. No. Wait on it, take your time, study these, and then attempt the exam. Once you get into the exam, you're going to see all the questions. There is no lockdown browser. However, because I just did this exam review, which I think you're all going to find very helpful, and which I think basically pinpointed every question, I'm going to change the time on the exam. Okay? I'm going to change it. There are 40 questions. I'm thinking about, let's see, probably 70 minutes, 75 minutes or so should be enough for 40 multiple choice questions for you to complete the exam. Another thing that we have is a extra credit opportunity. If you go to the discussion board, post a question, you get 0.25 points added to your exam. If you post a reply to a classmate's question, post an answer to a classmate's question, you get point 0.5. Maximum points that you can earn are is 3.25. Basically, that's one question. Each question on the exam is worth 3.25. So the extra credit is going to be 3.25. As far as curving the grades, we'll see how the class does. I believe you're all going to do a whole lot better than my previous classes because this was the first time that I have offered exam review session. And I'll need your feedback, guys, too. Shoot me an email, send me an email if you like the video and if you like the exam review and if you want me to keep it up for the next exam and the next exam, I'll be more than happy to. But if you think it's useless and you didn't find it helpful and you didn't think it affected your grade in any way, let me know so I can stop it and I won't post another video because all these our class is online. Our class, we are one of the colleges that we do not do hybrid. Everything is online. But I thought maybe this would be helpful to a lot of you. Anyways, good luck studying. Uh, Take notes. Rewatch the video if you need to. And thank you for your attention. Bye.